it's really critical for all these bioelectric states to ask who the interpreter is. So the mapping, right, much like with the DNA, the mapping between a distribution of voltage states and then some anatomical, state later on is really critical. And we spent a lot of time, and we're still thinking about. Originally, we thought that it might be specific voltage levels mapped to specific organs, and then we saw that that wasn't right, and then it really appears to be a pattern, differences across space. So what was all the interesting thing happens when certain cells of a particular voltage are sitting next to cells of a different voltage, and it's the difference that's actually meaningful to the outcome, not the absolute values of either side. It's the difference that matters. And so all of this brings to mind, okay, well, who's reading this? What's the interpretation machinery? So we have a few models now. We put out one, and there's one that's in revision right now, of asking how does a collection of cells read a spatially distributed bioelectric pattern and turn on specific genes as a consequence of this. So not an individual cell voltage turns on genes within that cell. That's kind of easy, and we found that years ago. there are 5 or 6 different transduction pathways that do it, but much more interesting is how do they recognize a pattern, and how do they know if the pattern is correct? How do they know what it means, and so on? And so that's what we're wrestling with now, and there's definitely an interpretation issue there. And so there are, we have a computational model of how that happens. It also, I think, ties into a very deep issue about memory, because there are these memory transfer experiments, right, where you transfer, somebody like David Glanzman might just transfer RNA from a trained animal into a naive brain. We've transferred pieces. So we'll do things like transfer pieces of an animal from one to another, and we look at propagation of morphological memory, propagation of behavioral memories, and so on. And you have the same issue with the decoding on the other end. So the thing that's always bothered me about, and it's not just for RNA, it's for any material substrate for memories, is that if I train an animal to some sort of weird relationship that never, sort of not evolutionarily prepared for. So, I don't know, 3 yellow light flashes means take 2 steps to your left, or otherwise you get shocked, right? So animals can learn this. So let's say that ends up being encoded in an engram of some kind of crazy molecular structure. Maybe it's an RNA, maybe it's something else. I take that, I shove it into a naive brain, and there's got to be a decoding mechanism that can look at that structure and go, oh, look at this hairpin turn. Oh, I see 3 yellow light flashes. Ah, right, got it. Well, where's that code book? And you can imagine something like that, something for something evolutionarily expected, like fear of the dark or something. We can all share the same code book because it's built in. But for these really arbitrary things that is not plausible that we share a code book that already pre-specifies that, it becomes really hard. And then the worst problem is, it's bad enough when you're moving it from body to body, but recall within the same body is the exact same issue. Because for me to read my engrams, I don't have access to the past. All I have is whatever was left, whatever traces were left in my brain and body, right? Maybe not even just the brain, but I have to interpret those. So 3 hours from now, I'm gonna have to interpret whatever was left by past me as a message to my future self. I have to do the exact same thing. I have to look at whatever this is and figure out what they mean, and just sort of on the fly keep rebuilding this. Yeah. So I think I don't know, Chris, what you think, but I think this interpretation machinery is really the key to all of this. There's another lens on this too, which Josh Bongard and I just put out a paper on polycomputing, which is this idea that the exact same set of physical events can be interpreted in different ways by different observers, and thus be doing different computations literally at the same time. And so we talked about he and his student Atusa have this amazing sort of mechanical example of multiple computations being done by the same piece of particulate matter. And I picked out a bunch of biological examples from that paper, where basically one way to the title which Josh came up with is And there's plenty of room right here, which goes back to like this idea of room at the bottom, right? Feynman's. And so this idea that in biology there isn't any room anywhere else because it's all packed full of stuff. There's stuff everywhere, and so the way to squeeze more out of it is to evolve additional observers who do interesting things with what is already going on, right? So as opposed to trying to tack on new mechanisms, and one thing that's interesting about that is that if what you're evolving is a new perspective on an existing set of events, it means that you don't risk breaking those events. That's completely different than trying to make tweaks and hope that you don't lose your past gains, right? You basically don't touch it's observation only, right? So you don't touch the thing that's going on. And as a simple example of that, we had this other paper recently where you take a gene regulatory network, and depending on how you look at it, meaning you pick 3 nodes and you call one of them the conditioned stimulus, one of them the unconditioned stimulus, and one of them is the response. If you pick the right nodes as your CS, US, and R, you can show that thing is doing associative learning, but only if that's your perspective on the system. If you have a different perspective, you'll see it doing something completely different. And in fact, multiple observers can have 2 different mappings, and they will see it. They will have 2 different pictures of what it's doing, but nobody's touching the actual network. You're not rewiring it. You're not changing synaptic weights for memory. You're not doing any of that stuff. It's literally what perspective you're taking as an observer. So, and there's many examples in biology. So that's another thing, right? You've got some kind of a encoding system, and it's working great for one thing. Pretty soon something will evolve which takes advantage of that just by interpreting that as a different kind of computation that you can make some sort of use out of, right? So that's kind of another aspect of this.
Yeah, in a sense, to put this in more computer sciencey language, this is what interpreters and compilers are doing, but particularly interpreters. Right, you have down at the machine level, or slightly above the machine level, you've got a whole bunch of processing going on, and layering a high-level language on top of it is layering an interpretation. It's assigning a different semantics to all of that processing that's already happening. And within that, using that semantics, one can interpret what's going on as, a Zoom call or something.
Yeah.
Whereas without that semantics, you have a completely different idea of what's happening.
Well, and look at how powerful that is, right? If you had a if you were running a software company or something, and you had a candidate that you were interviewing, and they say to you, well, I'm a reductionist. I don't, but I think there's no such thing as an algorithm. It's Maxwell's equations that govern what the electrons do. They just do what they're going to do. You'd never hire that person because they wouldn't be able to. They wouldn't code anything. It's hugely empowering to think that the algorithm makes the electrons dance, right? Because then you would go on and you would write things when you're thinking about it at that level. If you lack that abstraction, it's extremely hard to do anything. You've got the same batch of DNA. And so this in particular is our Xenobot example. You have the exact same batch of DNA. We don't change the genome, but depending on how that DNA ends up being interpreted, you end up with a frog embryo or you end up with a Xenobot. And it's the exact same, right? It's the exact same DNA that is somehow, what is it encoding, right? Well, it depends. It doesn't just encode a frog. Who knows what the heck else it, down here, who knows what else? So it's this idea of, I don't know, information or environment or something is sort of, is the prompt that gets this thing to generate. It's like a generative encoding of some sort, and there's a prompt that gets it to go in a particular direction, and you end up with something like this, or you end up with something like that. Well, yeah, right, and that's interesting too. Back to the memory thing. So I think the point there is that you can store multiple images sort of on the same piece of film and just recover them with different signals. There's an amazing book called Shuffle Brain, and it's this guy Paul Peach back in the 80s who did all these experiments in memory in salamander, and he basically was, he started out by looking for memory and taking out different pieces of the brain and showing them, well, it's actually everywhere, and then he would move the pieces around, and the salamander was, would basically be completely fine and do all the tasks and everything, and then he would move pieces from goldfish to a salamander, and the vegetarian would become a meat eater, and the meat eater would become a vegetarian. It's just amazing. But the second half of his book, so the first half of this book is all these experiments. The second half of his book is a holographic model of memory, where is sort of inspired by these experiments of non-locality, like he couldn't find it anywhere in any particular region and so on. He pulls out all these analogies of trying to store multiple different memories and whatever else is in there in literally the same hardware, right? They all have to overlap somehow. But this issue of where is it stored has been driving me nuts for a long time, because there are many such things when the distribution of primes and all the where did, like, that stuff doesn't seem to be it doesn't seem to depend on the physical facts of the universe. Like, where is all that? Or even just the thing where you've seen this Galton board. Yeah, it's just it's a piece of wood, and it's got a bunch of nails stuck into it. And you take a box of marbles and you dump it over, and they go boom, 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 and in the end you get this bell curve, right? You always get the bell curve. So now you could ask, where is the shape of this bell curve stored? So you start looking at the wood, and you look at the nails, and then you look at the distribution of the nails. None of those have anything with, none of them encode in the strict set. So where the heck does it come from, right? There's a million of these things.
Yeah. Why are there normal distributions?
Right, or truth tables, right? So you evolve. I like this too. You evolve a voltage gated ion channel, which is basically a voltage gated current conductance. It's basically a transistor. You have a couple of these things, you can make a gate. If you have a logic gate, you have a truth table. Where's this truth table? Like, you gain in the fact that the NAND is special and all this kind of stuff. Where is all that? It sure wasn't in your ion channel design. It's you get that for free somehow. It's like this incredible free gift from I don't know where it comes from.
Yeah. If I encode a particular reference frame that lets me identify a table, then I'm in effect stipulating what counts for me as a table.
Yeah. Okay. Oh, I see. So, okay, that's what.
And anything that fits those criteria is a table by stipulation.
Yeah.
For me.
Okay. Okay.
Maybe totally different for you.
So it's the policies of the people, cells, whoever it's going to be, that are replacing the components that makes this thing the same, because they need to execute their changes in a way that preserves some kind of invariant for them. So they're going to choose where to put the boards and everything in a way that preserves what they think of as the ship. That's the only way this is going to work. So again, we're back to this idea of observers, and starting off with the notion that if you want the thing to stay the same despite molecules come and go in the body, cells come and go. If you're a cognitive system, ideas come and go and mental states come and go, but you're going to stay the same. For you to stay the same, there has to be a replacement policy of some sort. And then that gives you the idea, that gives you the ability to do what you just said, which is better than staying the same, is actually a policy for changing you slowly into something else. So, if you're a caterpillar, you are maintained for a while, but eventually there's a new policy that actually maintains you and it turns you into a butterfly. And, are you the same? You've got some of the same memories and you've got some other stuff, but a lot of things have changed. But it's all, it's all about the policies that are not just keeping you the same, but actually slowly transitioning you to some future sort of representation of what you're going to be. And in order, and you to do that, you have to place the molecules, the cells, the information in the right places to be consistent. Yeah.
In this case, the aspect of the environment is the actual user of the representation that says, what gives this representation an identity condition for me is a particular utility. And as long as I alter the representation in a way that changes that utility only slowly, I can count it as the same thing. Otherwise, there'll be some dramatic failure. right? It loses its identity for me because it no longer does the job I need it to do.